Good afternoon. Thank you all for coming to the Arlen Richardson Student Research Day. This is an annual event where students in the Biology of Aging program at the University of Texas Health Science Center at San Antonio host a distinguished lecturer who has made remarkable contributions to science that have directly influenced our mission here at the Barshop Institute for Longevity and Aging Studies. This event is named for the founding director of the Barshop Institute, Dr. Arlen Richardson. Through his leadership, the Barshop Institute has become a world leader for studying the biology of aging. Although he has recently retired from his director position, his guidance will be sorely missed. His support of student development and individual research achievements is unmatched. Because of him and his devotion to student success, every student leaves here with the potential to become leaders in their field. There will definitely be big shoes to fill in the years to come. Thank you, Dr. Richardson, for everything you've done for students. On behalf of the students of the Biology of Aging program here at UTUSCA, we are pleased to welcome Nobel laureate Dr. Mario Capecchi to the Barshop Institute. Dr. Capecchi was born in Verona, Italy in 1937. Later, he came to the U.S. and went on to study chemistry and physics at Antioch College, where he received his Bachelor of Science degree. In his graduate studies, he went to Harvard and, under the guidance of the world-renowned scientist Dr. James D. Watson, obtained his Ph.D. in biophysics. As for his professional career, if we were to take the time to list all of his accomplishments and awards, we'd be leaving him with little time to speak. Dr. Capecchi is best known for his pioneering work on the creation of genetic techniques to specifically target and delete genes in mice. This work earned him a Nobel Prize in Physiology and Medicine in 2007. To say this work is important would be an incredible understatement. It has revolutionized the field and enabled us to make genetic models that mimic human diseases and has made us better able to understand the complexities of life. Many students here at the Barshop Institute utilize these genetic mouse models every day in order to understand the aging process. Without his work, much of the research we conduct here would not be possible. Without further ado, here is Dr. Mario Capecchi to discuss gene targeting into the 21st century, mouse models of human diseases from cancer to neuropsychiatric disorders.
So it's always a pleasure. I can be heard in the back, okay? Yes? It's always a pleasure to be a guest of the students. And they've been terrific hosts. They've done a terrific job, and you should be proud of yourselves. So what I'm going to do today is share with you two stories, just to give you a flavor of what the laboratory is doing and how we approach problems and hopefully provide some insight. Okay. So I want to first give also credit to the Barshops, and you're very fortunate to have such a benefactor, particularly at a time like this when NIH is not as easy to persuade giving you money as usual. So the first is cancer models. And the cancer models that we chose are sarcomas. And the question you might ask yourself, why sarcomas? They're not as common as carcinomas, but they have a number of features that are particularly of interest to myself, and they're shown here. First of all, they affect a critical population. Most cancer is a problem of old age. As we progress in age, the risk of getting cancer increases. Sarcomas, on the other hand, affect children and young adults. So pediatric oncologists, if that's what they see. So that's, and the second point is that they're extremely aggressive, and that's one of the reasons why you see them in children and young adults. And because they're not as common as carcinomas, they don't receive as much attention. So it's a niche opened up, and they also have a number of advantages. For a modeler, it's important to start at the beginning. And often in cancers, you end up with a tumor. You know many things have happened, but you don't know the order. And even with whether the order is critical, that is, if you have steps A, B, C, D, and so on, is it critical that they are in that order, or does it matter and could be any order? But in sarcomas, and I should also preface this by saying in biology, whenever we make generalizations, there are often exceptions. And what I'm going to be telling you all evening when I tell you something's happening, there will be exceptions. But on general, in terms of sarcomas, we think we know the initiating event. And that is the breakage of two chromosomes and then rejoining, but with opposite partners. So in terms of information content, everything's okay except at the junction point, where you're creating a new gene, which is half coming from one chromosome and the other half coming from the other chromosome. Okay? And this is common to 25 different sarcomas. Each specific has a specific different chromosomal translocation, and it's a reciprocal translocation. Okay? Then the final point is that they're not as complex as carcinomas. And the reason for that is that a hallmark of carcinomas is genome instability. And once you have genome instability, you have thousands upon thousands of events occurring in the cell. And it's very difficult to sort out what are the 5 or 6 players that are important for that cancer versus all the thousands of events that have occurred. In sarcomas, often the chromosomal translocation is the only abnormality that you can see. Okay? So that makes identifying steps, subsequent steps, that allow tumor progression to be identified much more readily. So those are the reasons we are working with sarcomas. The other thing I'm going to be saying is we, and it's always the royal we. Remember that the we is really students and postdoctoral fellows, and I'll give credit where credit is due. Okay. So the model that I'm going to be talking about is synovial sarcoma. Okay. And when we started, this is what was known about synovial sarcoma, and that is that they occur near joints. And that's where the name comes from, because synovial tissue is also near joints, but that's a misnomer. That is not a cell of origin for synovial sarcoma. The histology of synovial sarcoma is complex. There are 3 histological subtypes. There's monophasic, which is spindle cells, mesenchymal cells, biphasic, which also include epithelial cells as well as mesenchymal cells, and then there's poorly differentiated, which is common to a number of sarcomas, which are small cells with a big nuclei. So those are the histology. And this is the translocation. In this case, the translocation, and I should say in this case, we, is Malde Haldar, which is a very talented graduate student. The histology, the 2 genes are involved. One is on chromosome 18, and it's called SYT, and the other gene is on the X chromosome called SSX. SYT is also known as SS18, so in the literature you'll see either one or the other, and I'm going to be using both terminologies. And the genes that are involved are both chromatin remodelers. SYT is involved in complexes that are involved in activation of transcription. SSX is involved in complexes that inhibit activation or translation. Okay? And the fusion gene is shown in the next slide, and it includes almost the entire SYT gene, so has the activation domain, and then includes half of the SSX gene, and that half has the repressive domain. So this fusion gene is a schizophrenic molecule having both an activation domain and a repressive domain. Okay. Now, I should also point out that neither has a DNA binding domain. Okay? So it has to, the complex itself has to bind to some other protein, which then recognizes particular sequences. Now, this is the model, and the model is dependent on the properties of that particular cancer. In this case, we didn't have to worry about space. Okay? The space is being controlled by SYT, and it is ubiquitously expressed in every cell of the body. So we didn't have to worry about space. All we had to worry about is when and what cell type. Okay? And So we chose the Rosa locus because the Rosa locus is ubiquitously expressed from early embryogenesis all the way through adulthood. Into the Rosa locus, so Malay Haldar simply isolated, got a tumor, human tumor, isolated the RNA, made cDNA, and the first one he isolated happened to have SSX2. Okay, there are 9 SSX genes in humans, 11 in mice. SSX1 and SSX2 are commonly found, and there are a few examples of SSX4, and none of the other ones have ever been seen in the fusion gene. And so the one he isolated turned out to be SSX2, and this is the cDNA for that fusion gene. Okay? So we place that into the Rosa locus. We put an iris, which is a ribosomal reentry site, and then we put EGFP. So whenever this cassette is expressed, those cells will turn green and we can see them. Okay? And we put that in the Rosa locus, but between the promoter and this cassette, we put a stop sign. Okay? And we flank that with LOXP sites, and then this is a site-specific recombinase where in the presence of these LOXP sites, it mediates a recombination event between them, and in the process, if this is in the same orientation, excises that DNA, turns on now the promoter is driving formation of the fusion gene as well as making the cells green, and then by controlling where Cre is, we control where the fusion gene is produced, and also that these genes, those cells then turn green. So the first thing we asked was simply what happens if you form this fusion gene in every cell of the body. And that's HPRT-Cre. HPRT-Cre is expressed at the 2-cell stage. Every cell of the body is going to express it. And the consequences is shown here. And what you get, which is fortunate, is a green blob. Okay? All the 3 tissues for making the embryo are there, but they're completely disorganized. And so this embryo is quickly reabsorbed and does not produce cancer. But that tells us that our product is very cytotoxic with respect to development. Okay, so absolutely no development occurs. These mice do not develop tumors, but it does tell us something about what this product is doing, and it's bad things. Okay? So then we asked, where else should we look to see where you form tumors? And here we decided to look at muscle. And why muscle? For 2 reasons. One is that I showed you histologically there are mesenchymal cells there. Okay, so that may indicate a muscle origin. The other is that the other thing that's in common with joints is where the tumors always arise is also muscle, because you want to be able to move a joint. Okay? So that correlation said, well, maybe we should be looking at muscle. And so what you do is generate Cre drivers for all the whole muscle lineage and ask, when you activate the fusion gene in a particular lineage, do you or do not get tumors? Okay. So the first thing we looked at is muscle progenitor cells, PAX7 Cre and PAX3 Cre. Okay. So these 2 mice, so this is the mouse that has that construct that I showed you, okay, in the Rosa locus, and then this is a construct that contains the Cre driver that then specifies where during development is that fusion gene produced. And the consequence is, again, embryonic lethality and no tumors. Okay. So then we went to all the way at the end of the muscle lineage and used MIF6 Cre to ask, does fully differentiated muscle produce tumors? So, and that's here, MIF6 Cre. We did that experiment, and we got a surprise. And the surprise was a myopathy, and it's a classic myopathy. Okay, I'll show you that here. So here's normal tissue, longitudinally cross-section, and this is a muscle tissue in the presence of the fusion gene. So you can see smaller fibers, you can see wavy fibers, and you can see all the nuclei lined up. Okay? Now, you make muscle at 2 steps. You make it, you pattern the muscle during early embryogenesis from the somites. Okay? And then after that, you have the muscles all been patterned. Then you have the bulk of the muscle being made, lots of muscle. Okay? And that occurs in mice about 1 day before birth, in children about 2 weeks after birth, and that's when they start really making a lot of muscle. Okay? And that requires satellite cells. Okay? And and satellite cells are not only involved in making this massive muscle when the body needs a lot of muscle, but it's also included, for example, if I exercise too much and I destroy muscle tissue, then this system is used to regenerate that muscle. So it's involved in muscle regeneration. You destroy muscle cell fibers, and then you regenerate through satellite system. And the hallmark of that is all the nuclei lined up. Okay? And that tells us that now what's happening is the muscle tissue is dying and then being replenished through the satellite system. Okay? But they get myopathy, and then in about 6 months, 9 months, they actually die of the myopathy, but do not develop tumors. So that tells us that fully differentiated muscle itself is not a progenitor cell for this tumor. Okay? Now, this is interesting for another point of view, though. Chromosomal translocation, the frequency, both in the germline as well as in somatic tissue, is about 10 to the minus 3. Okay? Extremely common. In your lifetime, you have about 10 to the 17 cells. That means 10 to the 14th chromosomal translocations. In cancer, it's easy to recognize that a chromosomal translocation occurs because that clone of cells is expanded, and now we have a signal to look after. In somatic tissue, that's much more difficult. And 5 years ago, you had no way to detect it. Okay? Now, with deep sequencing, now we can see a 10th percent event, a 0.1% event, 1 in 1,000, 1 in 10,000. Okay? And what's interesting is that now people are starting to see chromosomal translocations in myopathies. Okay? But our prediction is that you're going to see it in a lot of other tissues. Okay? That's actually a way of having somatic mutations that can give rise, for example, in the brain. You could have all of a sudden one lineage having a translocation and affecting its behavior. Okay? So you're going to have somatic phenotypes that are going to be a result of translocation, and now we have the tools to start to seeing that, and indeed people are seeing it. And it's not surprising if it occurs fairly early during development, you're going to have a very large clone that has all of that at the somatic level, which would then be spontaneous, rather than being genetic. So anyway, so where do we go from there? So the next step in doing myelogenesis is actually myoblasts themselves. And here we had two choices. We could use MYF5 or MyoD. These two genes have overlapping functions. You knock out one, muscle's okay. You knock out the other, muscle's okay. You make a double mutant, and now you don't have any muscle. And you have deep trouble. You can't move. Okay? So Malay made MYF5-CRE, okay, and asked what happens there. And what he got was what looked like synovial sarcoma. Okay? We got tumors, 100% penetrance, always near joints. We look at them, they actually go all the way to metastasis, and the pattern of metastasis in the mouse is the same as the human pattern. Okay? And we think these are metastases simply because A, they're much smaller tumors, and B, they're in tissue that does not express MYF5. Okay, and here we're showing you in the lymph node. Oops, sorry. Come back. In the lymph node, and here's actually one occurring in the cerebellum. Okay. Boom. There are over. This is a histology. Remember that I told you it's complex. You can have monophasic, which are spindle cells, biphasics, which have actually both mesenchymal cells as well as epithelial cells. And that's what's shown here. Okay, so here it is essentially a biphasic tumor. Here's oops, sorry. Yeah, no, that's correct. And then here's monophasic tumor. Okay. Markers. There are over 25 markers that have been identified in synovial sarcoma. And here we're showing you epithelial markers. And then as well as myogenin is interesting. Okay, myogenin is a differentiated muscle marker, and every differentiated muscle marker is not apparent, is not expressed in the tumor. and that's why it was not recognized as a muscle tumor. BCL2, 98% of the tumors are highly expressed BCL2, vimentin, the mesenchymal marker, highly expressed in the tumor. And that's true of all 25 different markers that are specific to synovial sarcoma. Further, we've actually looked at expression profiling, and with respect to 1,000 genes, thousands of genes now, it has the same fingerprint as human synovial sarcoma. Okay? So we believe we have a legitimate model for synovial sarcoma. Here was a surprise. Okay, here's the tumor, it's green, and here's the tissue it's coming from. This is actually autofluorescence, and that's muscle, all muscle autofluorescence. Okay? But the muscle isn't green. Remember that when you make a myofiber, you essentially fuse thousands upon thousands of myoblasts all together to make the myofiber. Okay? Yet the muscle is not green, and yet we're labeling all MIF5 labeled cells. Okay? So that was sort of a surprise. Why? And the way to look at that is simply to look very early in embryogenesis when muscle is first being generated. Okay? So we're looking at 11 and a half days of gestation as the initial fibers are being formed. And what you can see, here's the fiber, and here are the myoblasts that are going to contribute to making the fiber. And what you can see is the green, the MIF5 label cells are there, but they're not participating in making the fiber itself. Why are they not participating? And the reason is they're dying massively, okay, by apoptosis. So having the fusion gene there is simply leading them to apoptosis, except wherever there's cartilage. So here's a rib, it's producing that's cartilage, and what you can see is all the green cells are still there, they're dividing, and something is being produced essentially by the cartilage that maintains those cells and allows them to go through cell divisions, and then accumulate all the subsequent events that allow then tumor progression. And we're very interested in trying to figure out what is that substance. The other thing with this puzzling is, remember that you make muscle by fusing all of these myoblasts. Okay? And I'm killing most of the myoblasts except wherever there happens to be cartilage. Okay? So how's this mouse running around? And I told you that there is overlap between essentially MyoD and MIF5. But redundancy can be of two types. You can have the same gene in the same cell and each be able to perform the same function. Alternatively, you could have independent lineages. You have a lineage being made by MIF5 myoblasts, a separate lineage that's making MyoD positive. And we can test that very simply by doing the simple trick. And that is go into the same Rosa loca, now put diphtheria toxin in that locus, again have a stop sign flanked by LOXP sites, and now drive it with MIF5. We'll kill all of the MIF5 lineage and ask, does these mice make muscle, and are they running around and the muscles functions? And that's shown here, and indeed that is true. Here is in the absence of Cre, we can see lots of labeling. Okay, the cells are turning green. Here is in the presence of Cre, we've killed all those cells, but there's still muscle there, or else you would have seen this big concave, because right there you would have seen absence of tissue. Okay? So that tells us essentially that they're independent lineages. And further, if we simply look at a time of, as a function of time, this is simply looking at MyoD and MIF5 lineage. What we're going to see right at this time when we're starting to make muscle, now the MIF5 lineage is starting to be dying off. And if we looked at a few days later, what you would see that lineage completely dead, whereas the MyoD lineage is taking off and thereby compensating for the loss of MIF5 lineage. And these muscles do indeed run around. They can develop synovial sarcoma, but they have functional muscle, saying that they are independent lineages and can compensate for each other. Okay, so in summary of this part, is simply I've showing you that muscle is a good candidate as the progenitor cell for this tumor, and that the properties of the mouse tumor are very, very similar by multiple criteria to the human material. So let's look at mechanism. That is, what is the fusion gene doing? Okay. And in this case, remember that both of these proteins don't bind to DNA, so we can't use that as an assay to pick out where is this particular protein involved. What we have to do is make essentially an IP, precipitate out the complex, and then ask who are the members in that complex. And this experiments were done in collaboration with Nielsen in Vancouver, University of British Columbia. And here is the gel, and we know the identity of all of these proteins, but I'm going to look at three. Okay, here's the fusion gene itself, that protein. This is ATF2, activation transcription factor 2, and TLE. These are both proteins that are involved, essentially, again, in chromatin remodeling. Okay? ATF2, activation transcription factor 2, is involved in activation domains, and the DNA binding domain is the cyclic AMP responsive elements. So wherever you want to physiologically up regulation of this gene in response to cyclic AMP, ATF2 is doing the job. Okay? TLE, on the other hand, is the opposite. That is, it's associated with repressive complexes and turns down the transcription of any complex that it's associated with, and particularly it's associated with the inhibitory complexes that I'll talk about in just a second. Okay. This simply shows that the fusion gene is required to bring these two proteins together in a complex. This is simply a glycerol gradient. At the bottom is sediments, big things, and then at the top is smaller and smaller things. And if you have a cell line that doesn't have the fusion gene, we put it in, and now we can show that they co-precipitate as a complex. And in the absence of the fusion gene, they separate their sediment independent of that complex. Okay. We can also make deletions essentially across the fusion gene and ask where do these members bind. And what we're showing you here is that ATF2 binds the amino-terminal complex of the fusion gene, whereas TLE, the repressive component, then binds the carboxy terminal of this fusion gene. We can also disrupt the complex by using interfering RNAs, and here we're showing you by simply using an interfering DNA to the fusion gene. In the absence of the control, the complex sediments at the bottom, if we deplete essentially the fusion gene, then now this complex breaks apart and the proteins sediment separately. And this is the model based on a lot of other work, plus what I've just shown you. And what it looks like is that essentially the fusion gene acts as a scaffold for bringing to ATF2 target genes this whole repressive complex, including histone deacetylases, the polycomb group complex itself, the inhibitory complex, and thereby turning off specifically ATF2 target genes. Of interest, for example, is EGR1, early growth factor 1, which is a tumor suppressor gene. And here we show that essentially both in cell lines which are human of origins, SY01 is a human cell line, synovial sarcomas, and then on the opposite panel we have cell lines developed in our mouse, tumor cell lines, and what we show is that if we interfere with formation of this complex, then the growth rate drops. Okay, so it's dependent on maintenance of growth. The complex is dependent on that. Okay, plating efficiency goes down if in the presence of the complex, and apoptosis goes up enormously in the presence, in the absence of the complex. And here we show you just a heat map. So green is low expression levels, red is high expression levels of 3 related sarcomas. And what you can see, and we're looking at essentially specific target genes of ATF2. And what you can see is the heat maps are very, very different in these 3 different different sarcomas. And here we're doing actually both in terms of immunoprecipitation as well as DNA sequencing, showing you that the complex sits right on top of the cyclic AMP responsive elements. That is the target of ATF2. And this has been repeated with multiple targets. In this case, it's EGR1 as a target gene. And if we interfere with the complex, what we can see is transcription of these target genes now goes back up again. So it's inhibited in the presence of complex and released from inhibition when you interfere with formation of the complex. Now, I showed you that part of the complex was histone deacetylases. So this argues that possibly inhibitors of histone deacetylases may interfere also with tumor formation and also disruption of the complex. And here we're looking at that. Romidepsin is a classic inhibitor of histone deacetylase inhibitor, and indeed, in the presence of the drug, we disrupt the complex. Okay? Similarly, if we look and where the disruption occurs, essentially is an interface between TLE and the fusion gene itself. And here we show that in the presence of the inhibitor, now transcription of these target genes goes back up again. Okay? So what that suggests is that this would be a good target, essentially a good drug to use against this tumor. And indeed now clinical trials are going on in 2 different sites to see whether deacetylase inhibitors is effective against this drug, against this tumor. Now, the other thing I pointed out is that 98% of the tumors express BCL2. And BCL2 has become a popular target, again, because it shows it in many tumors, for using essentially developed inhibitors of that. Okay, and this is the classic inhibitors, and they go against the BH3 domain of BCL2. Okay, and this is Navitoclax is commonly used. It's been used now in small cell lung cancer, and it's been used in a couple of lymphomas and so on. And they work temporarily, and then the tumor essentially reverses the effects of the drug, and then you have to use a different drug. And the reason that the tumors escape is what they do is they turn on other BCL2 members that do not have a BH3 epitope. Okay. However, with respect to synovial sarcoma, we made an interesting discovery. So the escape route is shown here. So normally these are the BCL2 members that have the BH3 domain and that the drug is effective, and these do not have a BH3 domain, and then they are turned on and then allow escape of the tumor from the effect of the drug. However, what we found is that in synovial sarcomas, that escape route is actually specifically turned down by the same route that I just showed you. Okay? So here we're showing you MCL1, and blue is low activity relative to red, which is high activity. And what you can see is in synovial sarcoma, the expression of those genes is down by the same mechanism that I just discussed. Okay? So that tells us that this drug might be particularly effective against synovial sarcoma. Okay? So we can test that. And we can test it in cell culture, and indeed the cells are very sensitive to BCL2 inhibitors. We can test it in conjunction with doxorubicin, which is the most commonly used drug against synovial sarcoma. And then the cells are really sensitive. And this is both human cells as well as mouse cells derived from the model. And then we can also look in vivo. This is actually a 2-week exposure to the drug. And what we can see is loss of mass of the tumor, and as well as, this is the tumor number, how many tumors per mouse, and this is the mass of the tumor. And again, we get a very effective loss. And as a consequence, now we're starting essentially trials, clinical trials, human clinical trials with respect to BCL2 inhibitor. Okay. So that's that part of the story. Now I'll move on to the next part of the story, and that is neuropsychiatric disorders. And you might think that a mouse is a poor model for our behavior, and I hope I can convince you otherwise. We got into this by serendipity. The lab has had a long-standing interest in Hox genes. Here he's showing you the Hox complex, and compared to the Drosophila complex, and what you can see is quadruplication and many, many Hox genes, 39. And we've knocked out all of these, and different students in the lab have had responsibility for different Hox genes. And Joy Greer, in this case, is we, and her responsibility was Hoxb8. So she knocked out Hoxb8, and she looks at it, and it looks pretty normal. So then she decided, well, I'll look at behavior. And she looked at many different behaviors, but one that was unexpected. First of all, that a Hox gene should affect behavior was sort of unexpected. But what she found was that grooming was affected. Okay? And all animals groom. And the syntax is pretty much the same. You go into the shower, you lather your hands, and you start washing your head. And from your head, you work down to your body. And if you had a tail, you would end up with a tail. Okay? And so that's what the syntax is. And that's quite reproducible and common to all mammals. But I should point out that also all animals groom. Right? Drosophila groom, coelegans groom. And you might ask how coelegans groom. Right? No hands, mama. Okay, but they do. And the syntax is the same. The syntax isn't affected. That is, the grooming pattern in these mice is perfectly normal. What's affected is the number of bouts and the duration of the bouts. So it's how much time are you spending grooming versus other behaviors. And what Joy did was very robust. She took infrared cameras and simply took movies of mice. And then, frame by frame, simply jotted down what is the mouse doing throughout a 24-hour period. Okay? And in this case, she's looking at eating behavior. Okay? And these are SIB pairs, and they're also matched for sex. Okay? So this would, say, be a female pair, and the black bar is the mutant, Hoxb8 mutant, whereas the gray bar is a wild-type littermate. Okay? And so they spend the same amount of time eating. She looked at many, many, many behaviors. Running around the cage, eating, drinking, building nests, running upside down in the cage. Anything she could think of, she measured. Everything was the same except for grooming. And these mutant mice spend twice as much time grooming as normal. Okay, and that's shown here. Okay. Further, we can induce grooming. You can spray water on a mouse, and that induces it to groom. And what we can see is an induced grooming assay. They're also spending twice as much time grooming as a wild-type sibling. Okay? This particular experiment was done completely on real wild-type mice, ones that you get out of the field, and we crossed into it the Hoxb8 mutation. We did this to show that, in essence, genetic background had no effect. So it's a robust phenotype independent of genetic background. Now, I told you a slight error. If you take 24-hour films and you do too much of one thing, you've got to take it away from something else. Okay? And what they take it away from is sleeping. So these mice spend the equivalent amount less sleeping. Okay? I'm going to show you a film that shows that the behavior is pathological. And so what you're going to see is the mouse is grooming. It grooms and grooms and grooms. And then when it turns, what you're going to see is that where it's grooming, it's actually removing the body hair. There. Okay? So it's actually, this is very similar to a human pathology. It's called an OCD spectrum disorder, obsessive compulsive disorder spectrum disorder, known as trichotillomania. And these patients compulsively remove their body hair, and they remove all body hair. This shows that it's pathological. They continue to groom after removing the body hair until they have lacerations. Okay? And this is similar to OCD, where patients actually wash their hands until they have lacerations. This is quite common. 3% of the population, and it's been looked at, all world populations have trichotillomania. 3% and independent 3% have OCD. These are not rare events. At this point, we have to put them into sleep. Now, this is a curious experiment. If you put a mutant mouse in the same cage, put a wild-type mouse in the same cage as a mutant mouse, it grooms it and pulls out its body hair, but the pattern is opposite. Okay? Before, I was showing you is removing the body hair for what we call the ventral side, the tummy side, and now on the other mouse, it's removing the body hair on the dorsal side, the back side, and that's what's exposed to it. And it chases it around on the cage and pulls out its hair. I've talked to hundreds of patients with trichotillomania. They have the compulsion of wanting to groom their other people. Okay? But they know, that's socially not acceptable. But the compulsion is there. Now, if this is true, okay, if it's able to groom another mouse, that tells me that it's not likely to be an itch. You're not likely to be able to feel an itch on another mouse. That is, it has something to do with our brain. And if that's true, it should be expressed in the brain. Hox genes stopped at when you were a crocodile. Okay? That is, it's very important for patterning the hindbrain, but it's not supposed to be expressed in the midbrain and the forebrain. Here I'm showing you that it actually is found to be expressed in the forebrain. These are adults, and what Joy did was to put all Hox genes are fairly simple. There's just 2 exons and an intron in between, and then regulatory regions around it. She put a nonsense codon in one exon and a LOXP site on the other exon. That made the message a little bigger, and she could recognize the mutant band relative to the wild-type band. Okay? So here are heterozygotes, have one copy of each. Here's a homozygous with only the mutant band. This also tells us this is not neurodegeneration because the signal strength is about the same. So the cells are there, and they're not performing appropriately, but at least they're there and doing something that gives rise to the aberrant behavior. So now we go to a different we. Okay, and this is Shakwon Chen, also a very talented graduate student. Now, taking movies and scoring them frame by frame is very robust, but it's a lot of work. Okay, enormous amount of work. 24 hours of movie that you're counting, and you do that with many, many mice. Okay? So now we switch to an automated system. These are platforms that are very, very sensitive to vibration. Okay, and as you walk around a platform, you give one type of vibration. If I'm running around it, I get a different type of vibration. If I'm drinking water, it's a different vibration. And scratching, grooming, you name it. Whatever motor activity you have gives rise to different vibration. And the computer can keep track of that and run as long as you want. This involves actually over 50 mice. And again, showing you that mutant mice spend twice as much time grooming as wild-type mice. Okay? But now that's an easy assay. Now we can expand that and allow us to do drug development and so on in these mice. Okay. Oops. Okay. Now I showed you that this gene is expressed in the brain, but in adults the expression level is low, and further it was salt and pepper, so it was difficult to identify which cells in the brain were actually expressing Hoxb8. And the way of getting around this is simply do lineage analysis. You get same locus, Rosa locus. You put GFP in that locus, again a stop sign flanked by LOXP site. Now we cross it with Hoxb8 Cre. Wherever Hoxb8 is expressed, those cells will turn green, and we can see it. Okay? And that's a very robust signal. And here we got an enormous surprise. It was just, couldn't believe it. The only green cells in the brain are microglia. And microglia are the immune cells of the brain. Okay? So what I'm telling you is that immune cells in the brain are controlling behavior. Okay? Because that's the only cells. So here is our green cells. Okay? Here's a pan-neuronal marker, CD11B, just labels microglia, and here you can see yellow cells, which are both green and red. What the other thing you can see is it's not all microglia. It's a subpopulation of microglia in the brain. There are actually two populations, one that does express Hoxb8, it's the only known marker that distinguishes into two populations, and the other doesn't. The one that doesn't, what we call the resident population, gets there very early in embryogenesis. Those cells are made in the primitive hematopoiesis and then come from the yolk sac, and as soon as the brain is formed, they're in the brain already. Okay? Whereas our microglia, as shown in the next slide, get in later, after birth. Okay, so here we're looking at green cells, and at P2, 2 days after birth, here's the choroid plexus. You start building up there. Here's the ventricular surface, and you start seeing cells being built there. And then by 2 weeks, now we have our green microglia all over the brain. Okay, and by 2 weeks, it's now steady state, and we can label those cells specifically with Hoxb8-Cre. Now, if what I've told you is correct, they should be expressed in the hematopoietic stem cells. Okay? So here we're taking blood and simply spreading it and looking at it in terms of different markers that distinguish myeloid cells from lymphoid cells, B cells and T cells, in the absence of Cre and in the presence of Cre. And what you can see is that whole population shifts in the presence of Cre, okay? Becoming fluorescent. So that tells us that they're either in very early progenitor cells or stem cells themselves. Consistent with being in stem cells, we look at two common markers that label stem cells, c-KIT and SCA1. This is a double labeled population. In the absence of Cre, in the presence of Cre, that whole population shifts, consistent with it being in Hoxb8 stem cells. In hematopoietic stem cells. Is there a phenotype with respect to Hoxb8? And here we had a little bit of difficulty because what we made was a Hoxb8 Cre driver that was an iris. Okay, and we specifically did that because we didn't want to kill the gene. Okay, but on the other hand, we cannot see wild-type cells because one allele will be always normal. Okay, so we see a 15% decrease, but this may be an underestimate if the other population compensates. Okay, we're knocking one down, the other might compensate, and therefore we only see a small change. So at least 15% or more less microglia in these brains. Now, this made a very strong prediction. We should be able to cure a mouse with a bone marrow transplant. And remember that Shaquan Chen said, please do a bone marrow transplant. He says, do a what? In a mouse? Okay, but he did it. But first of all, we simply want to see if we do a transplantation, do we get green cells in the brain? So this is simply using hematopoietic stem cells that are labeled green, put them in, and then ask if they get into the brain. We have to irradiate the mouse in order to get them into the brain. And we get very good engraftment. Okay, and at 4 weeks after, again, we see buildup in the choroid plexus, the ventricular surface, and then by 12 weeks after bone marrow transplant, now there's lots of green cells, and we get about 75% engraftment, so it's quite efficient. And here's the actual experiment. So this is a mutant brain, a mutant mouse, Hoxb8 mutant mouse. We irradiate it, lethally irradiate it, then introduce bone marrow, and then see what happens. 3 weeks after doing the experiment, we see the mouse removes its body hair. If I showed you this a week later, you see lots of lacerations. And this is 3 months after the bone marrow transplant. All the lacerations are healed, all the hair is back, and this mouse is permanently cured of that pathology. And if you look at how much time is spent grooming, here's a wild type, here's a mutant, and now it goes back to normal grooming behavior. Okay? Permanently cured, just by bone marrow transplant. We can do the opposite. We can put mutant bone marrow in a normal mouse and give it the pathology. So this says it's both necessary and sufficient for the pathology. But there is a problem. Okay, Hox genes have many functions. Remember that a given Hox gene is responsible for everything going on at that level. Okay? So many, many thousands of target genes, and they're doing lots of things. And one of the defects in Hoxb8 mutants is actually sensitivity to pain. In fact, another group in Belgium, published that our phenotype could be explained by this insensitivity to pain. Okay? So we had to look at that. So let me show you the phenotype first. So here is a spinal cord cross-section, and just using a pan-neuronal marker. And if you were judicious and looked very carefully, what you would see in the mutant, fewer cells right here in the dorsal aspect of the spinal cord, and more disorganization. But that can be seen much easier if you use markers. That's shown here. So GRP positive is labeling the incoming sensory neurons. And then calbindin and calretin labels the interneurons that receive those inputs. Okay? And what you can see in the mutants is fewer cells and more disorganization. There's not a nice laminar structure, essentially. And that's the mutant phenotype with respect to the spinal cord. We can also look at it behaviorally. That's shown here. And one way to measure this is simply to put a mouse on a hot plate and see how quickly it jumps. And if it has insensitivity to pain, it takes 3 times longer to react. And that's what you're measuring here. And it's a very robust, easily quantitated feature. Okay, so the mutants are insensitive to pain. Okay. So now we can ask, what's happening with respect to this phenotype? The first hint that told us that we could separate those two phenotypes was actually in the bone marrow transplant themselves. And that's shown here. So remember that the bone marrow transplant completely cured the mouse of the behavioral phenotype with respect to hair removal, okay, and lacerations. However, it has no effect on insensitivity to pain. Here's the wild type, here's the mutant, and here's the rescue, and still insensitive to pain. Okay? So that told us, wow, maybe we can separate those two phenotypes. And the way to do that is simply conditional. Knock it out in the bone marrow and see what happens. Knock it out in the spinal cord and see what happens. Okay? For the bone marrow, we use Ty2-Cre. And Ty2-Cre labels the hematopoietic stem cells, but also endothelial cells. Okay? First of all, it does label the microglia green, so that's good in the presence of that reporter gene. And here's the actual experiment. So we knock it out in the bone marrow as well as endothelial cells, not in the spinal cord, and we get the full phenotype. And that's shown here, hair removal, and if we look at how much time spent grooming, this mouse now spends twice as much time grooming, but it's not affected by the hot plate, that insensitivity to pain. So with respect to the bone marrow, we can separate those phenotypes. Okay? And we can look at the spinal cord. So that's shown here. And what we see, essentially, again, with those same markers, here's the mutant, here's the knocking it out in the bone marrow, and the pattern is the same as wild type rather than as mutant. Okay? So then, again, by that criteria, the spinal cord is okay by knocking it out in the bone marrow. Now we do the opposite. We knock it out in the spinal cord and ask what the consequences are. And for that, we use Hoxb8. And the reason is that both Hoxb8 and B8 are strongly expressed in the spinal cord. And the pattern is indistinguishable. And that's shown here. So if I showed you Hoxb8, it would be identical in the spinal cord. It labels a few microglia in the brain. Okay? But if you count how many microglia are labeled by Hoxb8 versus Hoxb8, it's a 15-fold difference. Okay? So it doesn't affect a lot of microglia. And here's the actual experiment. And here we see no removal of body hair. If we look at how much time spent grooming, they're identical to wild type. However, the now response insensitivity to pain is now recapitulated. Okay? So that has allowed us to separate it in the other direction. So that tells us that two phenomena are not interrelated with respect to phenotype, even though the same gene is responsible for both. So, and this shows you the same thing in the spinal cord. And again, what we can see is that now we recapitulate the mutant pattern rather than the wild type pattern by knocking it out in the spinal cord. So this raises a real question, and that is, how does microglia control circuitry in the brain? And one can imagine many scenarios. One thing that microglia do is simply produce cytokines, chemokines, and even transmitters, neurotransmitters. Glutamate, for example, is produced by microglia. So one way to talk to the neural circuit would be to produce these chemicals that somehow then the neurons themselves pick up and modulate their activity. The neural circuit has been defined in humans by essentially fMRI. You simply look at patients, and what you see is that these regions of the brains are overly active in OCD patients and also in trichotillomania patients, essentially from dorsal aspect of the basal ganglia to the motor cortex, prefrontal cortex, thalamus. And that's also the same circuit that's involved in learning repetitive behavior. Okay? So that's one possibility. Another intriguing possibility is these strange projections of microglia. They have these long filopodia that extend and extremely dynamic. I mean, they're moving around in space all the time. They extend them, contract them, wave them around in space. And you can calculate, because there are a lot of microglia, that they actually can they can feel, essentially, sense the entire parenchymal space in less than 1 hour. I mean, so they're simply all the time waving these things around. Then all of a sudden, what you see if you use two-photon microscopy is that they stop. Okay? And then what happens is that these filopodia go around a synapse. You can see that by EM electron microscopy, that this surface essentially is juxtaposed right across around the synapse. Okay, and the question is, why would you do that? I mean, that's a lot of energy and a lot of expended time to do that. And one possibility is that Dow's direct coupling, essentially electrophysiological coupling, between a microglia and a particular neuron, okay, at the synapse, and maybe be able to modulate that activity in some way, again, that nobody knows yet how to do that. But all the technology is there now to ask those kind of questions. Okay, you essentially, at Stanford, at MIT, people have developed ways of taking a channel that's susceptible to light. You can open it and close it with respect to light. Okay, and then and if it pumps sodium, then the cell's activated. If it pumps chloride, then it's silenced. Okay, that essentially is perfect for this kind of questions. We can activate microglia and ask whether neurons know it. We can activate neurons and ask whether neighboring microglia know it. You can transmit light in pulses. The reaction time is very short. It's less than milliseconds. As soon as you turn off the light, boom, it goes back to normal right away. Okay? So you can give essentially information, and then you can sense whether the other member can respond to that. Okay? And that would tell us there is direct communication and allow us to try to develop the methods to assess how that communication occurs. So those are the kind of questions now we can ask and see how microglia are affecting neural circuits. Now the other question you might ask is, what does Hoxb8 have to do with this phenotype in humans? Okay, all I've said so far is that they look like they have OCD. Okay, but we have a gene. And I also told you that 3% of the people have trichotillomania, so it's easy to get DNA. Now, the other question you might ask is that I'm talking about knocking out 2 copies. This is a recessive mutation. Okay? So obviously, 3% of the people are not going to be homozygous for any gene, hopefully. I mean, that would be a lot of inbreeding. So that's not seen. So a Hoxb8 homozygous is not going to be present in many populations. Okay, so that's the other. So, and the other thing is 3% of the people have it. Okay, how can you account for that by a genetic model like this? And there are two possibilities. A lot of genes give out the same output, or there's a very common polymorphism in the population. Turns out that Hoxb8, there is a common polymorphism. 20% of the people carry it. And if you follow the mutant phenotype, the polymorphism, the mutant polymorphism, it tracks with the phenotype. That is, they have trichotillomania. So that now closes the circle. That is, we're not simply looking at similar behavior, but possibly similar causality in humans and mice. Okay? And so that's what we're following up next. And I'll leave you one more teaser. Okay, and that's over 600,000 bone marrow transplants have been done worldwide at this point. 3% of the population. A lot of overlap. So the question is, when you get a bone marrow transplant, you get lots more than the bone marrow transplant. Okay? That is, normally they're trying to cure cancer or something that's going to be killing you. And so I've been calling all of these different bone marrow transplant places and ask, have you seen anything bizarre happening with respect to behavior? And they say, no, we haven't been looking at that. Okay? I have a pile of emails about this high from patients that say, wow, lots of funny things have been happening. And it can go both ways. Okay? The other thing is, I don't think that this whole system was set up to control grooming behavior and aberrant grooming behavior. Okay? That just doesn't make sense to me. So what are we really looking at? Okay? If you look at any human psychiatric disorder, monopolar depression, bipolar depression, autism, schizophrenia, even Alzheimer's, long been known, 30 years of literature, that there's an association between those behavioral disorders and the immune system. That's been known a very long time, but nobody knew cause and effect. Are you depressed and therefore your immune system isn't working well? Or is your immune system defective and the risk of getting depression is increased? Okay, who's cause and effect? In this model, we know cause and effect. Defective microglia give rise to defective behavior. Okay? So we have a model essentially to make that connection and now start to study what that interaction is between the immune system and behavioral output. So with that, I'll close and open it up for questions. Thank you for your patience. Oh, yeah. So I'd be happy to entertain any questions, and the wilder, the better. We'll go start in the middle.
Yeah, so in terms of microglia, the phenotype, when you lose Hoxb8, what really changes with microglia? There is definitely literature about
what microglia are doing. Yeah. And that is what we're just following up. I mean, initially we couldn't do that experiment. And the reason we couldn't do that experiment is that we use an iris Cree. Okay, and so one allele will always be wild type. And then, so we never could see how the mutants behaving, the mutant cells. And now we've created a new allele where we have the Cree inserted right in the middle of the coding regions. And now that will allow us to do those kind of experiments. And then, and there are many possibilities. I mean, microglia certainly are involved in, if you get an infection, they're there, they go there and kill the pathogen. I mean, they are macrophages, essentially, of the brain. If you have a stroke, they clean up the mess, okay? Alzheimer's is a little trickier, okay? Some people say it helps, and other people say it doesn't help, and probably both are right. That is, it's the same thing as being chronic, okay, inflammation. Initially, the immune system's doing good things, but if you make it chronic, now it does bad things. Okay? So I think that's going to be true of many neuropathologies. You're going to have both windows, essentially, of opportunity for these cells. And the question is, how do the Hoxb8 microglia behave relative to, what we do know is that they're different in the sense that 60% of the cells cannot compensate for a Hoxb8 mutation in 40% of the cells. That says functionally they're doing something different. The other slide that I'll go over this. This is curious because maybe complete coincidence. This is all the inputs coming, these are projections coming from the spinal cord. And I showed you that it strongly expresses Hoxb8. So neglect all of this red. Okay? But if we look at this and blow it up, what you'll see is red cells, which are Hoxb8, and green cells, which are the resident population. Okay? So we can distinguish those two populations. And what's curious is this is a circuit that's been identified essentially as the OCD circuit. They're high in Hoxb8 expressing microglia, whereas in other areas of the brain, now that population is low. Okay? So coincidentally, wherever the Hoxb8 happen to be high is also in the area of the circuit that's involved in this behavior output. Okay? So that tells us, that these cells must know where they are and know where to go, because they can migrate. I mean, if I put a needle here, what you'll find is all of these microglia go to that needle, any destruction of tissue, and massive migration of these microglia. They become little amoeboids and just travel like crazy. Okay, and the question is again, do the Hoxb8 do the same thing, or do they do something different? And now we have the tools to be able to address that. But a year from now, I'll know the answer. So.
Well, you mentioned that there were two populations of microglia. The majority were derived from Neuropsychiatric, that came earlier, and then about 1 or 2 weeks postnatally, the Hoxb8 was a smaller population went into the brain.
Right.
Is there a developmental functional integration as to when the Hoxb8 cells populate the brain and when circuits are established?
So did everybody hear the question, or should I repeat it? In the back, you heard it? I mean, what he's asking essentially is, when does this population get in, and is this related essentially to the function of the circuit? And one other thing that actually microglia are involved in, which has been, again, established by other investigators, is they're also involved in synaptic pruning. Okay? You make twice as many connections as you need, and then you wipe out half of them, and microglia are the things that do it. Okay? So that's another possibility. In terms of the question, what we see is that this second population gets in at P0, starts getting in at P0, and then by 2 weeks after birth, then they essentially set up a steady state. Okay? There's an interesting correlation with 2 weeks. Okay? And 2 weeks is also when astrocytes now close off the brain-barrier barrier. Okay? That's when they send out their feet, and then they essentially now stabilize the blood-brain barrier, and after that, nobody gets in. Okay? Or lots of things don't get in after that. Okay? And microglia may be one of those. That is, that unless you have pathology, we radiated, okay, and that we needed the radiation in order for engraftment in the adult mouse. Okay? Without radiation, those cells don't get into the brain. And we also needed it because we want to create a hole. Okay? We want to wipe out the system it already has so that now you get engraftment. Okay? So for both of those reasons, we did radiate, and in the absence of radiation, nothing happens. So it looks like that's the second population is essentially sweeping in essentially from birth to about 2 weeks, and that is also the period when you're doing a lot of synaptic pruning. Okay, that's at the same time that you just madly, and what you see if you actually follow the amount of microglia in the brain, it's actually sweeping up just ahead of that, and then after that it reaches it drops down and then reaches a new plateau level, and that's steady state then. Yes.
The population of people that have the 20% of polymorphism.
Yeah. Yeah.
Are they of any particular racial distribution?
No, it's been found the same in all racial distributions, pretty much the same throughout the world. And I should say, it's not in the coding region. Okay. So we don't know what this polymorphism is doing. Okay. But what we know is it's a polymorphism that's traveling with the phenotype. However, if I looked at it and had to guess, what it looks like is a splicing defect. Okay. And further, what we can do now is simply regenerate that exact polymorphism in a mouse and then ask what are the consequences. And the prediction would be, and we're just doing those experiments, that you will see reduction in Hoxb8 activity.
But in the body, hematopoietic compartment. Do you see any favorite distributions, cell distributions? Is there some selective factor that's pushing that 20%?
Yes. And I'll answer that. I'll give you a long answer. First of all, Hoxb8 is involved in hematopoiesis. That's why it's there, okay? And what's doing is exactly this. That is, it's distinguishing lymphoid cells from myeloid cells. It's important in making that switch. And so it has a role in hematopoiesis itself. Okay, the other question is, I'll give you an extension of that. Remember I told you that I don't believe 3% of the people are homozygous with the Hoxb8 mutation. Okay, that's not possible. But what is possible is that you have your heterozygote, you have that polymorphism, and then you have something else coupled to that that would be equivalent of having lost 2 copies of Hoxb8. What is something else? You know, if I was a betting man, I would say it's genes evolved in the hematopoietic system. Okay, so those together then would give you the phenotype in humans. Yes.
It's very interesting the way you look for genetic background effects, which are, widespread by using an actual wild mouse. Do you do that frequently, or was that a one-off thing? Because it's a dirty secret of aging research that we don't know anything about aging in mice, only black 6 mice.
That's right. No, and we did it because I have a neighbor that has a scientific neighbor, not just a real neighbor. Well, they're real neighbors too. That has a barn where they're doing lots of experiments. A set-up building where they do lots of experiments with wild-type mice, and they were interested, for example, in how our Hoxb8 mutants would compete with their wild-type mice and all that kind of stuff. So it was all set up so we could do those crosses. And it's not something I recommend because, domestic mice that you are really domestic. Wild-type mice are really wild. And you're just maintaining them in a cage. As soon as you open the cage, whoop, they're gone. So we wanted to do it simply because we wanted to prove that the genetic background. And I should point out that those really are important experiments, particularly with respect to behavior. It's actually it's amazing that any inbred mouse can walk around. There's no other species that nobody has been able to cross 20 generations and still have that species be alive. Okay? And mice, they can do that. But as a consequence, every inbred mouse is different because you've trapped a different constellation, essentially, of mutants in that inbred mouse. And it's not surprising that genetic background varies enormously with respect to different inbred strains. And where that output would really be, behavior still is complex. Another startling thing by this work is that you mutate a single gene and you get out a very specific behavior. That's pretty bizarre. I should say, okay, did I say that? I don't think. Did I say why I think the system works? The reason is what's in common between all disorders, neuropsychiatric disorders, and that is stress. Okay. If you look, I've asked patients with trichotillomania, when do they have their activity? And it's when they're stressful. There's always an absolute correlation, level of stress and output of behavior, whether it's anything you want to name, depression, whatever it is. Okay. So I think what this system is really doing is controlling the level of stress. And that's what it's made to do. And our output is what we could measure is trichotillomania. But that's not the normal output. And it'll be different depending in humans, depending what other variation of genes your parents happen to give you. Okay, so sometimes it'll be schizophrenia, sometimes it'll be axiomatic. But that's the commonality. And I think, and it's very important to control stress. At times you want to be stressful. Tiger's chasing you, you run and run. Okay, that's a good response. If you want to communicate with people, then you want to lower that stress level. And it's it doesn't have to be immediate. It can take minutes, or it's not something that has to be instantaneous. but you want to calm yourself down so you can do things under rational conditions. So I think that's what this system is doing. And in the absence of that, now these mice, and they are, I mean, the other phenotype that these mice have are, we've looked at 4 independent behaviorals. It's easy to measure stress by all sorts of behavioral tests, and they are very stressed animals. So I think that's what's really
So that begs a question.
Okay.
Whether the microglia are affecting the neurons are actually sensing the microglia.
Yeah.
So
now, who's talking to who and how? No, I think that's an open question. But we can do it either way. I mean, we can activate the microglia and ask whether neurons know about it, or we can do the opposite. We can activate specific neurons and ask whether neighboring microglia react.
But if there's a general rule in sensory systems that you use whatever piece of information you can,
oh yeah, sure.
Then if the microglia are going to respond to a stressful state, then you could well imagine that the brain evolves a mechanism to monitor the microglia and adjust accordingly.
Sure. No, and I think sometimes we are, we're sort of neurocentric. We think neurons do everything, and I think I'm of the opinion that there's a lot of astrocytes that we. Sometimes they're producing the neurotransmitter that then the neurons use to propagate their signal. I mean, so there's I think, who's doing what, and you could even argue that neurons are simply cables, okay? They're just transmitting information, and the brain is actually, microglia and astrocytes and so I don't really espouse to that, but,
you don't want to do that
that's
in
the
public,
opposite extreme. But I think we do take a neuron-centric approach, and I think it's important to point out that communication can be both ways, and in one direction sometimes, in any other direction other times. Yeah.
Any more questions?
I wanted to ask you about the Grooming mouse, which has the spike mutation.
Yep.
And that also
Yep.
is a model of
Yep, that's right. Yep. That's right. So that's part of the that's right. That's right. Right. That's right. No, I think there are now 3 mutations in the neural circuit, and what's always in common is that the dorsal aspect of the striatum is involved. That's where the neural defects are, and so that's a very important focal point. But it's also, when you look at gene families, it's those members that are expressed in that region that then give rise to the phenotype. And so that defines essentially the neural circuit. It's consistent with it. And the phenotype is actually very similar to what we're seeing. It's essentially excessive grooming leading to pathology. An interesting question will be in humans, how much of that is going to be as a function of defects in the neural circuit, and how much is going to be a defect in microglia. And I think that's approachable now.
No more questions? Okay. Thank you, everyone, for attending.